You hear me? Okay. So we're gonna do the same rules as previously. Everyone's gonna come one by one, and I will raise the hand when it's 4 minutes, and stand up when it's like the end of the talk, like 5 minutes, and it's better to conclude at that moment. So we're gonna start by the first person, who is Luis Cali. Do you prefer PowerPoint or PDF? Okay. It's here. Yes, if you want. Otherwise, there's one. Yes, this is laser. This is as you want. Let you start.
Thank you. Today we learn about a lot of very interesting subjects, history, chemistry, but now is the time we get to the real subject, goats, and how do goats' weight varies. So let's start by get very simple. One way to imagine the variations of goat's weight is to divide the variation in two groups. First, you have growth that give this kind of trend, and second, you have the uterus load that give this cyclical variation. Because goats are farm animals, so they tend to have babies every year or so, which explain this cyclical variation. So we just divided the variation into two groups, so why not divide it into four? And that's exactly what the GROW model is doing. We talked about growth, we talked about uterus load, we just have to add body reserve and maternal investment during lactation, and you get the whole model. Now the only thing left to modelize the weight variation is a way to manage those variation depending on the period the goat is living. So first the goat grow, and then the cyclical variation starts. So yeah, the other thing the models do is taking this kind of data and it make a weight profile for the goat, and it helps us compare the weight profiles between one goat and another. But as you can see, some data are missing around the birthing events, and this is a problem to adjust the model. And so we need the data, but we can't have it because goat skipper have something else to do when the goats are giving birth. So we need the data, but we can't have it. So that's where I try to help. My work was to analyze the database about kids' weight and to modelize their weight at birth. So let's start with the first basic approach, basic quantitative genetics. And let's start with a simple linear regression. So here you have the R squared. What does it tell? It tells us that barely 9% of the kids' weight variation over here is explainable with the dam's weight variation over here. So that's not a lot. But we get a low p-value as well. So what does it tell? Basically, it explains nothing, but it explains it very well. So we can't ignore it. But obviously, we need more parameters. So here are some of them. Sexes, males and females tend to not have the same weight. Or for example, litter size, if you're alone in the womb, or if you have 4 brothers and sisters, so you won't weigh the same. So here are the effects that will be taken in account for the model. For the breed, I've just tested it by making 2 different models, and so let's see about that. Here's the first model made using the data for alpine goats breed. And so what does it do? It just draw a line and adjust the line heights depending on the parameters. And visually, it looks like that. And on the right here, you have the calibration errors, and it's pretty nice because it's neatly distributed around 0. So it just shows that this model is a non-biased estimator. So that's pretty good. So now does it work for another breed of goats? Of course not. As you can see, it is overshoot by 1 kilo. So now we need to make some choices. Here are some of them. And here is the one I choose. So let's do everything once again. This was the code I used, and this is the result. It looked difficult, but really it just gives us the coefficient values for the next model. And so the next model is follow exactly the same principle, but with different values. So it looks like that, and the error is this time neatly distributed around 0. It is non-biased. So to summarize, here are the two models made to try to help the group model. And here is the whole presentation in one slide. And to open the subject, my models aren't very precise, so we need to continue the work in the future. Thank you very much.
Thank you for your presentation. So now we're gonna hear Jules Andrevon-Canut, sorry.
So, hi everyone. I'm here to talk to you about my research project, which takes place in the Central Supérex Research Program under the supervision of Jean-Michel Gillet at the SPMS laboratory, which is called Mapping Molecules in New Space. Can atoms be separated in velocity space? So, it was upside down. Okay, I'm sorry about that. Let's continue. So let's start with a simple analogy. Let's say you're in a mountain range with two mountains and you want to know which mountain you're sitting on. To do that, you can look at a topographical map which represents the height of the mountain, and you can see that naturally a path is taking shape between the two mountains. Well, the same question arises for molecules. You see, quantum physics tells us that once in a molecule, atoms are not precisely defined anymore, just like mountains are not precisely defined in a mountain range. So, for example, here we have the topographical map of the water molecule, which represents its electron density. And in the 90s, the physicist Richard Bader told us that if you want to split your molecule into different atoms, you must look at the gradient of the electron density, which are the lines perpendicular to the contours of the electron density. If you look only at those lines, just like for the mountains, you will see paths that will take shape naturally between the atoms. And if you do the same thing in 3 dimensions, you're able to split your molecule in 3 different regions, each region representing one atom. Okay? So now let's get back to our analogy, and let's say there are rabbits on your mountain, and you want to know about their speed. So you can look at the overall distribution of the speed of the rabbits, and you'll have something like that, which tells you that rabbits are going between 0 and 20 kilometers per hour. But what about the contribution of each mountain? This doesn't tell you if those rabbits are going faster than those of the other mountain. Well, this is exactly the question we're asking, not for rabbits in mountains, but for electrons in molecules. You see, we know the overall distribution of the velocity of the electrons, but we don't know the contribution of each atom separately. To do that, we need to take a step back. At the moment, we've looked a bit at velocities, we've looked at position, but what if we look at both at the same time? This higher dimensional space, which gathers both positions and velocities, is called the phase space. And for a quantum system, this phase space is represented by the Wigner's function of the system. So here, for example, you have the Wigner's function of the water molecule. An interesting property about this Wigner's function is that if you integrate over all the velocities, you obtain the distribution in positions. And if you integrate over all the positions, you obtain the distribution in velocity space. And why is that interesting? Because if you remember well, at the beginning, I said that we were able to split the molecule into different regions. So what if instead of integrating over all the positions, we only integrate over the different regions? So for example, we take the first hydrogen and we integrate over the position of the first hydrogen. Then we'll obtain the distribution in velocity only for the first hydrogen. Then if we do the same for the oxygen and finally the second hydrogen, we'll obtain 3 different distribution in velocity, 1 for each atom. And this is exactly what we are going to try to do. At the moment, we're not there. I've been trying to prove that the Wigner's function, which is essential to the project that I was computing, was correct. To do that, I first checked that it was respecting the known properties of the Wigner's function. So as I told, if I integrate over the velocities, I'm supposed to find back the distribution in position. So that's what we have here for the hydrogen. In blue and green, we have the exact solution. And in orange, we have the solution found by integrating our Wigner's function over all the velocities. And as we can see, the curves are very well fitting. Here's the same example for another atom, which is the neon atom, a bigger atom. And as we can see, it's working pretty well too. The second test was to compare to the results of the scientific literature. And so on your right, you have the Wigner function of lithium hydrate from an article of 1983. And on your left, you have the function I computed myself. And as you can see, the two figures are kind of close. So to conclude, I think our Wigner function is getting pretty good, and we are going to be able soon enough to proceed to the integration in each region of the molecule in order to understand the contribution of each atom in the velocity space. Thank you very much for listening. I hope you enjoyed, and if you have any question, feel free to ask.
Thank you very much for your presentation. Now we're gonna hear Nathan Chevalier.
So hi everyone. I'm going to present you my master's thesis, which is about the impact of conflicts on the trade of rare earth elements intensive products, and which was tutored by Daniel Mirza, who is an economics professor at Université de Tours. So to begin, rare earth elements are a group of metals that are particularly convoluted for their unique properties. And in fact, the production of these elements is mostly owned by China, as you can see on this graph. But simultaneously, China is engaged in a non-physical conflict against Taiwan since the 50s and the end of the Chinese Civil War. Okay, so the questions we ask ourselves is, first, which products are intensive in rare earth? So looking at these graphs from NKWT and Van der Waal, we understand very easily that at the world level, the most important sector for rare earth allocations is in fact rare earth magnets that are further used, for example, in high quantities in electric vehicles for their motors. Looking at trade data, we also identify that China is a very dominant actor on this market, exporting around 85% of the rare earth magnets in the world. So this is the equation we tried to estimate. In fact, in this equation, we estimate only the betas here. This is a gravity equation of trade, okay? And it links exports to characteristics proper to country pairs, so exports pairs between countries, so the distance between them. The conflict variable is constructed with the Google Trends website, which gives us interest quotations for specific quotes searched on the web by the people. So what we assume here is that the more interest people have for a specific conflict, and in fact the China-Taiwan conflict, then the more it should be intense at the given period. Then we consider a variable that identifies countries that have publicly announced their support for Taiwan, a variable that identifies when China is exporting the rare earth magnets, and we add, so these are parameters, so econometric parameters, so I won't go deeper in details, but this gathers, for example, variables that can influence trade, so that are characteristics from the countries that influence trade, but that are not linked to the conflict, okay? And our main parameter of interest is beta 6 here, which corresponds to the effect of the conflict, so here, on Chinese exports of rare earth magnets to countries that have publicly announced their support for Taiwan, okay? So coming to our results, we first observe a decrease in Chinese exports of rare earth magnets towards countries that have publicly announced their support for Taiwan. So that can be a reaction from China or from those countries that decide to show their support for Taiwan, not to export anymore, to import anymore from China, or both effects at the same time. But what is very surprising, for other countries that haven't taken side in the conflicts, we observe an increase in rare earth magnets exports from China towards these countries. So that could be explained as a precaution behavior. For example, countries seeing that there are tensions between China and Taiwan can fear not being able to import more magnets in the future and decide to export more now. And also, we decided to construct the same conflict variable for the Russia-Ukraine conflict to compare the impact of both conflicts on the exports and to see if the impact we observe is really due to the China-Taiwan conflict. Okay, and this project is going to be continued in the second year of master within the PhD track of Université Paris-Saclay. Thank you for listening and feel free to ask questions.
Thank you very much for your presentation. Now we're gonna hear, sorry if I don't say well the name, Tawakali Adul. Okay, sorry. But your name is Tawakali. Okay. So I'm gonna put your
Hello everyone, I am Tawakalit Damlola Habdaliakin and I am currently an M2 intern at the Institut de Chimie Physique, Université Paris-Saclay. I will be presenting my master thesis focused on the radiolytic synthesis of metallic nanoparticles stabilized by thermosensitive polymers and applications. This thesis project is supervised by Professor Isabel Lamp and Director Indra Meter. It's not moving. It's okay. Okay. Even this is not working. Yeah, it's okay. To start with, metallic nanoparticles are small objects composed of metal atoms with dimension between the range of 1 to 100 nanometer. To explain this nanometric dimension, a strand of human hair is about 1,000 times larger than these metallic nanoparticles, indicating that they are extremely small. And common metallic nanoparticles, but are not limited to this, include gold, silver, platinum, and palladium. And the versatility of this metallic nanoparticle results from their unique properties, which differs from their bulk counterpart. These properties are influenced by their shape, size, composition, and structure, as well as their assembly. For instance, to differentiate between the bulk metals and the metal nanoparticles, a large piece of gold have a golden color, as we all know, but when this gold is in form of nanoparticles, they exhibit a different color due to their difference in the interactions with light. This metallic nanoparticles has been utilized since antiquity, even when they are not identified as such, for their optical properties. A famous example is the light gyroscope, which changes color in the presence of light due to the presence of silver and gold nanoparticles. Similarly, the stained glass window of Saint-Chapelle in Paris contain the nanoparticles of silver and copper, which gives them the yellow and the red colors respectively. And also, the color changing properties of these metallic nanoparticles are influenced by their composition as well as the shapes they adopt, as seen on the image. And nowadays, these metallic nanoparticles serve diverse purposes in various fields. Some of the applications involve serving as antibacterial agent for this device designed with nanoparticles for prevention of infection, as well as catalysts for depollution, such as in catalytic converters for combustion engines. Now to talk about the current issues, in which one of it is industrial pollutions. Ecological balance are currently being disrupted by the residues emitted as a byproduct from advanced industries. These residues, when discharged in they causes environmental pollution when discharged in air, water, soil, and nature. And the increase in pollution has been linked to an increase in diseases, posing significant threat to human life through when individuals inhale, ingest, or come in contact with these pollutants. And common industrial residues, but are not limited to this, include industrial effluent, organic compounds, particulate matters, toxic gases, and oil spillage. These harmful pollutants can be converted to non-toxic pollutants with the help of a nanocatalyst, such as the metallic nanoparticles. And these metallic nanoparticles would serve as a catalyst, accelerating the rate of reduction or conversion of these toxic pollutants into less toxic ones. These metallic nanoparticles have been demonstrated to be efficient catalysts for depollution due to their small size, coupled with their large surface areas, their active size, as well as their shapes. So, to synthesize metallic nanoparticles of controlled size, shapes, as well as to ensure non-toxicity for pollution remediation, efficient and grease synthesis method, as well as effective stabilization method is required. Among the available methods is the use of ionizing radiation that are of high energy. And this ionizing radiation, when you use this ionizing radiation, no chemical reducing agent is required. The reducing agents are generated from the solvent, which in this case is water. And the absence of this chemical reducing agent eliminates the production of harmful byproducts, which could adversely affect human health and ecosystem. With this method, metallic nanoparticles are formed from the reduction of metal ions to metal atoms in the presence of the stabilizer, with the reducing agent generated from the solvent due to the effect of ionizing radiation. This is followed by the growth of this metal atom to form stable metal nanoparticles. This internship study aims to utilize a radiolytic synthesis method as well as the use of stabilization method based on thermosensitive polymer, which becomes soluble and insoluble below and above its temperature of about 32 degrees Celsius. We have utilized these metallic nanoparticles for catalytic reduction of a toxic pollutant, as indicated from the decreasing spectra as well as the color change. And lastly, the goal of this internship is to synthesize smart materials by combining the stimuli responsive properties of this polymer as well as the physicochemical properties of these nanomaterials for use in environmental remediation. Thank you so much for your attention.
Thank you very much for your presentation. Now we're going to hear Kenza Adjar.
So hi everybody, my name is Kenza Adjar. I'm a first year master student in biology and health at Paris-Saclay University. And today I'll be talking about my ongoing project, which is being conducted at Paris-Saclay Institute of Neurosciences under the supervision of Jean-Pierre Levroux and Cynthia Frock. So the title of my project is Microglial and Type 1 Interferon Signaling Implications on Brain Development and Behavior. So let's dive in. What's microglia? So microglia is a type of glial cells, which are cells that help neurons in the brain. And these are specifically the immune cells of the brain. They have a phagocytosis ability, which helps them keep the central nervous system homeostasis, but they also have a role in brain development. What's type 1 interferon? Well, interferons are cytokines, and cytokines are proteins that can activate and organize the immune response. And type 1 interferons are the primary antiviral cytokine. They also have a role in synaptic plasticity as well as learning. And there are 5 subtypes of type 1 EFN in mammals, and 4 of them in zebrafish, so Phi1, Phi2, Phi3, and Phi4. And why am I talking about zebrafish? Well, because it's the model that we're using in our lab. And there are many perks for using zebrafish because it's a vertebrate and genetically it's really close to humans. We can easily modify its genome. It develops quickly. They have transparent embryos, as you can see on the photo. And they are just the best model in the world, according to our fish facility manager, and she's really persuasive. For my project, we're using 2 types of mutated lines of zebrafish. So the first one doesn't have microglia, and the second one doesn't have type 1 EFN, which allows us to study the impact of these 2 on the brain development and on the behavior on 3 stages of zebrafish, larvae, juvenile, and adults. So how do we study zebrafish behavior? So for the larvae, we use well plates and a zebra box, which is equipped with a high-resolution camera and an infrared light. That allows us to track its movement and measure the distance and the time of its movement. So we can use either classical wells or wells that are divided into black and white. And with this, we can study the locomotion, the tigmotaxis, which is the tendency of fish to swim close to the walls and avoid central spaces, which can be a symptom of stress or anxiety. We can also study the place preference or DAC avoidance, which is also used to assess stress and anxiety, but also preference. And for adults, we can do all of that, but also study social preference. But the thing is, because they are big, we don't use plates like this, we use just tanks. So this is an example of an experiment that we did. And here's a video. I don't know if it works, but I don't know if you can see, but there's larvae that is just moving. And on the left bottom, you can see the layout of the tracking. And after that, we can do some statistical tests to see if there's a difference. In here, we're seeing the distance in the center of the movement of the larvae between the fish that doesn't have microglia and the control fish. And for this this specific experiment, we didn't see any difference in tigmotaxis. So now, how do we study brain structure? We use immunohistochemistry and confocal imaging. And immunohistochemistry allows us to target specific proteins that are expressed in the cells that we're interested in. So in our study, it's microglia. So we're using antibodies that are targeted specifically against a protein that's only expressed in microglia, and also acetylated tubulin, which is a marker of the white matter of the brain. And the antibodies that we use have a fluorochrome, which is a fluorescent molecule that allows us to see it under some microscopes, such as confocal microscopy. And this is an example of what we can see with the confocal microscope. It's really beautiful, and it's in 3D. So now you know everything about my project, and it's still ongoing, so hopefully we'll still have some results to analyze. Thank you for your attention.
Thank you very much for your presentation. Now we're gonna hear Oussama Zekri.
So hello everyone, Oussama Zekri. So this is my project consistent aerobond compressed faster, realized last year in Japan. Okay, so it's math. It's true that it's a bit different than what we have seen this morning, but let's dive into it. So I will talk you about a mathematical object called convex set. So what is a convex set? This is a set that contains some number of points, and when you pick two random points of this set and you draw the straight line that connect them, the whole straight line is in the set. So for example, this Pac-Man right there is not convex, all right? And in mathematics, convex sets are really important because we are often interested in projecting points onto sets. Like, for example, you have this point, you wanted to project it on this set, so you find the closest point that belongs to the set, PC. But when you have non-convex set, you are not sure that there is a unique projection. For example, in this example, there are 2 points that are closest to the point X. So that's why convex sets are really important. And the problem that I studied is called the convex feasibility problem. So you take a collection of M convex sets, you have a starting point in some space, and you wanna try to find this area here, the intersection of this M space. So for example, there you have 3 space. You wanna find a point starting from there that belongs to this. So maybe the first idea that comes to your mind is to decide to project alternatively. So it's sad because this was an animation, but it's not working. Maybe bad lecture, but never mind. You have this drawing, so you can just decide to project alternatively and hope that it will converge onto one point of the intersection. And this guy, von Neumann, made our dream come true because he proved that in the convex case, you can, so you have the convergence onto this intersection. And maybe a more sophisticated algorithm was invented by these two masterminds, Douglas and Rashford, and they decided not to directly project onto the set, but to take partial projection. So the animation doesn't work also, but there is this pretty hard to understand drawing, but what you just have to understand is that it's a way more faster than the previous idea to just alternate projection. And my work was to try to analyze the speed of this convergence and to compare it to the first idea. So with my colleagues, we invented this mathematical function, so the consistent error band function, that helps us to analyze mathematically so the speed of the convergence of the Douglas-Rashford algorithm. So if you have an algorithm that verifies these two assumptions, and it's the case for Douglas-Rashford, that's what I proved in my work. If you have this algorithm who verifies these two hypotheses, you have this theorem. So this is my first theorem, so I'm very proud of it, but it's a bit ugly like this. But thanks to this theorem, we have the information of the speed of convergence of the Douglas-Rashford algorithm. And maybe you will tell me, but what's the point? What it is used for? The answer is just here. So remember that an image is just a collection of pixels. So an image could be seen as a convex set. And if, for example, you have a collection of images that are blurred like this, you can apply this kind of algorithm to de-blur this kind of image. And I'm sure you don't know that your phone lies when you are taking a photo. When you are taking a photo, your phone doesn't take one photo. It takes a bunch of 10 photos and of 10 blurred photos, and it computes the projection, and it shows you this de-blurred image. And so my theorem, so a theorem that we proved, showed that with the second algorithm, the Douglas-Rushford algorithm, so the red one, it's way more faster. So you have just to use 50 iteration to be very precise, whereas with the first ID, it's really slow. So I'm done, and thank you for listening. So maybe a little bit of advertising. Just follow me on Twitter. Don't worry, I follow back. I post this kind of animation. So you didn't see the animations. That's really sad, but I will put them on Twitter. So if you follow, you will see.
Thank you very much. So now we're going to see a little video. So it's this one. So you can follow him on Twitter. Sorry. So now we're gonna hear Alexandre Clausel.
Okay. Good afternoon. I will present you my master internship work about a new tracking device for translation and rotation tracking in for neuroparticles embedded in neurons. So in your neurons, you have little molecules like that basically they work on your neurons, and they do step of 10 nanometers every 10 microseconds. And their goal is to keep your brain alive, basically. They bring the proteins, they get rid of the waste, etc. So why are we interested in these molecules? It's because it has been shown that in very early stage of disease like Alzheimer or Parkinson, the intraneuronal transport was disturbed. So a better understanding of the elementary actor of this transport will give lots of information about this disease. But to study these molecules, we have to be very precise, we have to be very fast, and we want to see the biological samples that are in 3 dimensions and with a lot of scattering of light, so it's really difficult. And instead of doing classical microscopy, where you take pictures and you, after pictures after pictures, you follow your particle. What we do is we put a single emitter that will be carried by the molecule, and we follow only this one. So we choose to use nanoparticles of about 100 nanometer diameter, and that have the particularity to do second harmonic generation, which is a nonlinear optical process that converts 2 photons into 1 with a double frequency. So the frequency is the color. Basically, you send red light and you get blue light at the end, and with the amount of blue light you have, you know at which distance you are from your particle. Now we need something to make our laser follow the particle, and for that we use a digital micromirror device, which is just a set of very small mirror of 10 microns, and you can switch on or off your mirrors, and by changing the pattern and carefully choosing the pattern, you can move in the 3 dimension your laser focus. And with that, we do 9 points around our particle, and we see which point gives the most intensity. That was for the position. Now for the orientation, we take advantages of another property of second harmonic generation, which is that it depends a lot on both the orientation of the crystal of the particle and on the polarization of the light. So here you see, if you are vertical polarization with a 55-degree angle, you have lots of emission. Instead, if you have 0 degree, you have almost nothing. So now we can try to we have to test if we are effectively able to follow our particle, and for that we put our sample on a little rotating stage, and we make it do turns. So when it turns, you see that at the same time the orientation will turn, and we did it, and effectively we had a cycle of about 30 microns, and in color you can see the angle between 0 and 2 and 180. So when we said to do alpha pulse, alpha turn, we see alpha turn, so it works. And we test it at different speed, and we show that we can follow up to 1,000 degrees per second, which is the limits of our motor. So maybe we can be faster, but we just don't know. And to conclude, now we started to do experiment on neurons, and we get a few trajectories with the angle at the same time, but we are still analyzing the data, so to be continued. Thank you.
Thank you for your presentation. Now we're gonna hear Mariam Ahmed Karim.
Est-ce que le pointeur marche ou non? Oui. Et là, c'est les flèches pour le... Donc avant, après, c'est ça. Tac, et du coup, hop, ça marche. So, hello everyone, I'm Mariam Ahmed Karim, and today I'll briefly introduce you my M2 Immunology internship that I'm doing with the Professor Géraldine Schlettlouf in the unit 996 of INSERM. And this is a project on the control of dendritic cell migration, identifying the molecular link between glucocorticoids and calcium homeostasis. So first of all, to provide you some context, so dendritic cells are immune cells that act like antigens in our tissues, and they are able to capture antigen and migrate to our proximal lymphoid organ to present the capture antigen to other immune cells for initiation of our immune response. Thus, their function is mediated by their ability to migrate. So previous studies in my lab have described a protein, a glucocorticoid response protein, to be involved in dendritic cell migration. So indeed, it was shown that when the gene coding for GILs was invalidated, the migration of dendritic cells were impaired. Also, it's widely described in the literature that calcium ions through the calcium channel ORI1 are going to mediate cell migration. So preliminary experiments have been realized in my lab where when GILs, the gene coding for GILs is deleted, calcium entry and the calcium channel ORI1 localization was impaired. So this brings us to the question that what's the link between the glucocorticoids and calcium signaling. So this brings to my internship. So the aim of my internship would be to describe the potential link between GILs and calcium response by studying a candidate protein, so SGK1 for serum glucocorticoid induced kinase 1. So it has been shown in the literature that GILs interacts with SGK1, stabilizing it. And in other studies, it was shown that SGK1 will prevent the degradation of ORI1, and thus it's going to regulate the calcium entry in the cells. So this was shown in renal cells, but I'm not working on renal cells, I'm working on dendritic cells. So I shall have to validate this model in my cells. And to do so, I'll be working on bone marrow-derived dendritic cells that are going to be either wild type or dendritic cells invalidated for the gene coding for GILs. I'll be treating those cells with different pharmacological modulators, such as glucocorticoids to induce SGK1 or even GILs or even SGK1. And I'll also be using an inhibitor of SGK1, and then I'll be quantifying the DNA expression level by quantifying the messenger RNA. I'll be checking I'll be doing the quantification of the protein level of the respective proteins, so GILs, SGK1, and ORI1, and I'll be looking at their localization using microscopy as well. So to conclude, my internship is here to characterize a glucocorticoid-dependent regulation of calcium ions that is going to promote dendritic cell migration. So this should follow, if ideally everything goes well, it should follow a model like this where glucocorticoid levels are going to regulate calcium ions, thus modulate the migration of dendritic cells. That's all for me. Thank you for your attention.
Thank you very much for your presentation. Now we're going to hear Mina Chapon.
Okay, can I start? Do you hear me? Yes. So, hi everyone, my name is Mina, and today I will talk to you about the covariant formulation of electromagnetomechanics coupling in the framework of relativity. So first, to give you a little context, I will redefine each terms of my subject so you can understand a bit more. So on the first hand, electromagnetism is the study of phenomenon between electric currents or fields and magnetic fields. And on the other side, mechanics is the description of matter through cohesion, motion and forces. So when you talk about a coupling, it means that each physics phenomenon will interact with each other. So for example, we call piezoelectric materials for which the electric field is linked to the deformation and the stress and vice versa. For example, they can be used in sensor, and on the other hand, magnetostriction is quite the same phenomenon but for magnetism instead of electricity. Now I'm going to explain to you why do I study such coupling coming from civil engineering. So in civil engineering we focus a lot on damage, so it consists in the study of initiation and propagation of cracks which then lead to the ruin of the structure. And even if we try to prevent them and to proceed to maintenance, we try to describe damage as much as possible. So by studying this coupling, if I describe the mechanical stress, I will be able to describe damage on the one side, and on the other side, by studying the electromagnetic stress, I will be able to describe the cohesion of matter at small scales, because it's the forces which link particles together. And so in this presentation, I will try to quickly explain to you how do we determine them. I'm going to need two tools, which I'm going to explain to you quickly. So on the left, you can see that I use a Lagrangian, which is a quantity representing the physics phenomena. And so this is a quantity which varies along a path, which I call C here. And this quantity has to verify the least action principle. It means that it has to be minimal along the path C. For example, when you throw a ball in the air, it will follow a parabola. So it means that the parabola is the path which minimizes the Lagrangian of the phenomenon instead, for example, of a wavy trajectory which would not really be realistic. On the other hand, I will work in the framework of general relativity because because I will work in a four-dimensional space-time, and in this framework, the gravitation is linked to the curvature of the space-time, so mass and energy deform the fabric of space-time instead of just being a force which links us to the Earth or big objects. And I will use differential geometry because it's the right mathematical objects to describe this. So now I will give you a little more detail about my work, and we will try to determine the stress. So as I explained to you before, we are going to use a Lagrangian. It will depend on our three main phenomena. So these are the gravitational and inertial properties, then the electromagnetic field, and finally the matter. So each one has a term, G, A, and psi. Then this Lagrangian will be described by a sum. So one term for each phenomena, so it's quite simple. We could add more terms, but for now it's enough. As I said earlier, it will have to verify the least action principle. So we will try to minimize this quantity along the path G, so it's the path of gravitation and inertial properties. And by doing this, we will obtain a new equation, which is the Einstein equation. And this equation puts into play two objects, the Einstein tensor, which describes gravitational aspects, and then the stress energy tensor. So in this new tensor, we have the impulsion, the energy, and the stress. So it's a nice tool, and inside we can find the stress. So we've done our job, and we've found both mechanical and electromagnetic stress in order to describe damage. And I would like to thank you for your attention.
Thank you for your presentation. Now we're going to hear Aizamal Aitsanova. Aitsanova? Sorry. It's Adjana.
Hello everyone. So I am an M2 student in the genetics, and my internship projects relies on the hypermutability of Mycobacterium tuberculosis. So you all probably know the disease called tuberculosis. It is caused by an agent, Mycobacterium tuberculosis bacteria, and this disease actually takes 1/3 of the world. It is a very widespread disease. And once it gets inside the host, human for example, it might get through different factors, including the antibiotics. And you know that it is very dangerous to misuse antibiotics. We can actually define them by the first-line drugs and the second-line drugs. And the second-line drugs are getting into the treatment if we see that the first-line drugs don't work. And the reason why they don't work is the antibiotic resistance. So when the antibiotic treatment affects the Mycobacterium inside our body, it goes through the DNA damage, and the accumulation of DNA damage leads to the mutations. There is a group of genes called 3R genes. It is a genes involved in the defense of the DNA of the bacterium. And if these genes get mutated, the chance of getting antibiotic resistance increase significantly because it basically becomes hypermutable. It mutates in a very rapid pace, which is why nowadays scientists are trying to find the new development of the antibiotics. To do that, they use genome-wide association study. Usually it applied on the human system. However, the novel science shows that it's possible to also apply it on bacteria. So the GWAS can be divided by the source of association into allele counting, the most classical one, the one you would see when you study the disease of a human, such as cancer or Alzheimer's. This is the main one. It doesn't take into account the evolution of the human or any organism of interest. However, the second type is a homoplasy counting. Homoplasy is a convergent mutations, the mutations that would occur at the same time in the taxa that would be distantly related or unrelated at all. And here we would take into account the mutations. The gray lines are the mutations happening in the phylogenetic tree across the evolution of the organism. So the GWAS would actually count the occurrences of mutations in order to find the association between the gene of interest and the phenotype. In our case, the phenotype of interest is the antibiotic resistance in tuberculosis, in Mycobacterium tuberculosis. So the basic pipeline of any homoplasy counting GWAS is based on the counting of homoplasies at each locus, so in the whole genome map. Then we calculate the empirical distribution of the homoplasies, and based on this distribution, we create a null data set where we make a randomized resampling, similar to the multiple resampling testing. And then after the estimation of the evolution, we predict the evolution that might have happened in the Mycobacterium tuberculosis and create an association testing. And based on this association testing, we can actually detect the mutations that might be connected to rapid antibiotic resistance and thus create, for example, new developing treatment or antibiotics that would be more successful in treating severe multi-drug resistant types of Mycobacterium tuberculosis. So the reason why we study homoplasy counting GWAS in the first place is because, first of all, it deals with many factors that disturb the association because of the microbial complications coming to the genome. I will not talk about this. So first of all, I've benchmarked my method on the data set that has many different strains across the world. And after application, we actually found some mutations. We used 2 different methods. It's called Putin, called after the Canadian food, and Trivas. And actually, we saw that the Putin is more sensitive. The asterisk shows the important mutations, one that actually are candidates for the antibiotic resistance compared to Trivas. So now we plan to apply these methods on our dataset of 50,000 genomes, 50,000 sequences, in order to identify the novel mutations that might be connected to the rapid antibiotic resistance that may lay into the future of the antibiotic development. So, this is Putin. Thank you.
Thank you for your presentation. Now we're gonna hear. Oh, I don't have the other side of the page. Sorry. Gabriela da Cunha Bradão Reis. So, here is your presentation.
Hello. Okay, good afternoon, everyone. My name is Gabriela Reis. I am a STAPS M2 EOPS student from Université de Paris-Saclay. And today I'm going to be presenting you my internship research topic, which is the impact of a motivational running app, effects on adherence and middle-term performance. This internship is funded by the Graduate School of Sports, Movement and Human Factors, and it is held at the Laboratoire Interdisciplinaire des Sciences du Numérique. My advisors are Professor Jean-Claude Martin and Professor Javier Sanchez. So my research has an interdisciplinary approach, blending psychology, computer science, and sports performance in order to analyze the effects of a personalized mobile app on running, motivation, and performance. And the World Health Organization really emphasizes the importance of physical activity to reduce health risks. However, despite its benefits, there are still many adults that remain inactive. So one possible solution to that would be using personalized mobile apps that utilize persuasive technology, which is specifically designed to influence behavior and improve engagement. So we have many psychological theories that can help us understand motivation in the context of physical activity. And one of them is the self-determination theory, which is very commonly used in this field. And it basically distinguishes between intrinsic motivation, which is based on internal satisfaction, and extrinsic motivation, which is based on external rewards. And it's important to notice that intrinsic motivation is the most stable form of motivation. So within this theory, we have the basic psychological needs theory that basically postulates that all humans have three essential needs, which are autonomy, based on which is basically feeling in control, competence, which is feeling able and capable, and relatedness, which is basically feeling connected to others. So when we have an environment that satisfies all these essential needs, we are able to see increases in well-being, performance, learning, and ultimately activity engagement, which is our goal. So researchers are also using the regulatory focus theory to tailor this intervention to the participants' motivational profiles. And it basically divides into promotion focus, where we focus on seeking gains, and prevention focus, which is focused on avoiding losses. So for example, a message for promotion, we would say, to maximize your chances of improving your performance, you should do this. While in prevention, we would say to avoid having a poor performance, you should do that. And we believe that by tailoring the motivational messages to the participants' regulatory focus, we're going to be able to enhance engagement. So in comparison to other apps on the market, our aim is to access the motivational impact and performance outcomes of a tailored mobile app, which is specifically tailored to regulatory focus. And we believe that we're going to be able to see increase in engagement in running, improve performance, and also enhance adherence to long-term physical activity. So this is still an ongoing study, so we are recruiting 15 to 30 recreational runners, especially if anyone wants to be a participant, talk to me at the end. And these participants will do pre and post intervention questionnaires to analyze psychological traits, as well as performance tests where we want to analyze aerobic fitness, explosive power, and biomechanical analysis. After that, they will start an 8-week intervention period where they will have to wear a smartwatch and use a motivational app that was developed by our research group. And this application will send these messages that will be tailored to the regulatory focus of the participants. The features of the app are based on theories, psychological theories, and examples are the collective challenge and the chat that is based on the relatedness of the basic psychological needs theory. So in conclusion, we present with our project innovative approach to enhance motivation and performance outcomes through this personalized application. And we believe that by integrating the psychological theories with this mobile app technology, we will be able to help individuals achieve their running goals and ultimately improve overall health and well-being. These are my references, and thank you for your attention.
Thank you for your presentation. Now we're going to hear Jean-Paul Nora. You want the PDF or the PowerPoint? So here it's to go before, here it's to go at the next slide, and this is for the laser.
Thank you. Thank you all for staying until the end. So my presentation will be titled Towards Atomic Boson Sampling Using a Bose-Einstein Condensate, which is a part of my ongoing internship, M2 internship. Don't worry, I will be explaining what's all that. First, I will be explaining what is boson sampling using photons, which is what people use right now. Then what we are trying to do, which is atomic boson sampling. And then how we are trying to do it experimentally. I think the best way to imagine what is boson sampling is to imagine a Galton board. I think we've all seen that toy where we input some balls, we let them fall and random walk because of obstacles, and then carrying them in boxes at the output. Each time we do that, we are sampling a given distribution, which is a normal distribution here. But we have different occupations each time we do sampling. Boson sampling is the same, but instead of using balls, we use bosons, which is a type of particles. Photons, the particles of light, are bosons. How we do that, we start with inputting some squeezed photons. Squeezing is a quantum feature that I will not explain, but is important here. Using fibers, we input a lot of photons. We let them interfere in a complex coupling layer of interferometers, and we measure the output with photonic detectors. The question that is raised is, what is the distribution at the output? Well, it turns out it's not an easy task to do. Actually, it is impossible. It is an impossible problem to solve on a classical computer. This is due to two quantum effects: the interference part, used because of the beam splitters we use in the coupling layers, and the squeezing part, the thing that I've talked about here. Squeezing is a squeezing of the quantum state. So it's a quantum feature that I will not comment on. But it is actually easy to sample on a quantum hardware. So if you do that in the lab, it's just easy to do sample after sample. Actually, a team in China have done that, and why we do that is to demonstrate that quantum hardware can solve problems that are not solvable on a classical computer, and this is to demonstrate quantum advantage. What they did is that they did sampling for around 200 seconds, and they estimate that it will take a supercomputer 2.5 billion years to replicate the same thing. My quest in this internship was to ask, can we do the same but using atoms instead of photons? Well, actually, if we start with a gas of atoms randomly moving, independently moving, and on the energy scale, you see that the atoms occupy the high energy levels. And if we reduce the temperature with techniques like laser cooling, we can arrive at a point where all the atoms occupy the lowest energy level and they start acting as coherent waves. This is what we call a Bose-Einstein condensate, or a BEC for short. But actually what we can do is tune interactions between particles and let two particles interact, and what will happen is that some excited states will get occupied, and that's what we call the depletion of the gas. And actually this is a source of squeezing. So what we are interested in is this part of the gas, which is the depletion part. So can we do boson sampling? Do we have the second condition, the interference one? Well, actually what we have naturally in our gas is two atom interference only. And this is natural in the gas. We don't have to put any beam splitters like we do in photons. But you might say that this is boring because you only have two atoms interference, but you said that you need a complex interference layers to have some advantage. Well, you're totally correct. But what we have in the experiment right now is what we are trying to do is to control this and to see whether the distribution at the output replicates the theoretical distribution, and then, in principle, one can increase the number of interferences and the complexity in the coupling layers, which will be the second part. I will not tell you about the results. The results are not conclusive yet, but I will tell you how we do that in the experiment. The experimental realization consists of first producing a Bose-Einstein condensate in a trap, and then removing the trap, let the atoms fall, and detecting them one by one on an atom detector inside boxes. Each atom will trigger some electrical signal, and we know that the atom is there. So we can see one by one. So this should remind you of the Galton board, where balls fall, but instead of balls, you have something that is 1 billion times smaller. But the thing is that how do we separate the Bose-Einstein condensate from the depletion, which is the source of the squeezing, which is what we are interested in? Well, actually, in the trap, what we have is a periodic potential, where the atoms see like some periodic hills, and they live on the valleys of this periodic thing. And as I told you that the atoms are actually waves, coherent waves, and I don't know if you know, but a wave that is passing through a periodic potential or periodic grating will diffract. I have a diffraction grating here. I don't know if you can see. But the same will happen with the atoms. This I stole from the lab today. And what we see actually in the detector is this pattern. So we see clusters, and those clusters correspond to the BEC, which are the coherent waves. They will diffract, and the rest of the boxes correspond to the depletion. So my part in the M2 in this internship was to look at those specific boxes and to compute their distribution and to compare this distribution to the theoretical distribution that you have. So thank you very much for your attention. This is the team behind all that, and if you have any questions, I'm available.
Thank you very much for your presentation. And we have finished the session. So if you want to eat downstairs, there's a lot of things to eat. And there will be some prizes given to some candidates at the end of the Congress. Thank you.